In the past, if you were told your child had cystic fibrosis, a genetic disease, it was a life sentence. But with the advancement of technology today, doctors have found ways to regulate the progression of disease and manage pain with medications. But what if it could be solved at the source and no child was born with genetic diseases? This is where, in the near future, CRISPR-Cas9 will come in.
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat. There have been other gene editing technologies, including ZFN and TALENs, but CRISPR edits small portions of the genome at a time, can be used for all organisms, and is much cheaper than other technology.
It starts with an inaccurate piece of DNA that needs to be altered. The CRISPR system contains two parts. The first is the guide RNA, which tells the nuclease where to go. Then there is Cas9. It is an enzyme that will envelop the DNA at a specific location and open it up. It cleaves or cuts the DNA in what is known as a double-strand break. Now Cas9 gives the DNA a new segment to be used to copy and insert into the genome.
So what? Where has this all come from? In 1953, James Watson and Francis Crick first discovered the structure of DNA. And then in the 1980s, the ability to edit a genome was first seen in prokaryotes, but it's said to be hundreds of millions of years old. Bacteria uses editing as a defense mechanism to fight viruses. In the early 2000s, food scientists at a Danish yogurt company realized that this was happening and turning their culture sour.
There are three main inventors of this technology: Jennifer Doudna, a biologist at University of California, Berkeley; Feng Zhang, a biomedical engineer at the Broad Institute of Harvard and MIT; and Emmanuel Carpentier, who is at the Max Planck Institute in Berlin. In 2012, Doudna and Zhang tweaked CRISPR to make it user-friendly and show that human DNA could now be cut at specific places of their choice. This makes it possible to add snippets of genetic material, delete parts of DNA, and even insert entirely new pieces of code.
There are many arguments over the ethics of gene editing. CRISPR could allow parents to choose the characteristics of their children. They could then choose eye color, height, and even predispose them for athletic, musical, and academic talents. There are also many concerns that can be raised when editing sex cells because it is possible that editing one part of the genome could affect generations to come.
At UPenn, researchers are studying CRISPR and how it can change genetic diseases forever. If the experiments provide successful results, then it is possible that genetic diseases such as sickle cell anemia, cystic fibrosis, and chronic conditions like type 2 diabetes and Alzheimer's could be treated and eliminated using CRISPR. There are also high hopes to be able to treat muscular dystrophy and genetic blindness.
Specifically at UPenn, doctors are running an experimental trial with cancer patients using the CRISPR technology. The team of doctors will take out some of the patient's T cells and use CRISPR to edit the genes in those cells. This will hopefully transform these cells into defensive cells and help the patient's immune system fight the cancer. The researchers will then inject the cancer-fighting T cells back into the patients to determine if they are now able to seek and destroy cancerous tumors. The experiment has only been done with 18 test subjects, so much more testing must be done before it can become a mainstream treatment option.
Right now, there are few setbacks before CRISPR can become widely used. The National Institute of Health, by far the world's largest sponsor of scientific research, will not fund studies using CRISPR on human embryos, germline cells. Also, many traits do not come from just one section of DNA, and each individual genome is unique in certain ways, making it difficult to target specific traits. So for now, scientists are focusing on somatic cell study.
In 2016, CRISPR was used to edit the genes of vegetables, sheep, mosquitoes, and all kinds of cell samples in labs. Other applications include creating blight-resistant crops, foods that have longer shelf lives, and comically muscular dogs.
As research is being done, many people in the science community are still worried about the ethics of CRISPR manipulating the genome. What people do not understand is we are already using technology that CRISPR is capable of in other aspects of science. For example, in vitro fertilization. Only recently has IVF become an accepted method to obtain a family. It was previously referred to as test tube babies because people are able to choose which gametes to inject. But as it has become more prevalent in society, people have accepted it. Frankly, they do not even think about it anymore. As time passes, as shown through the IVF technology, CRISPR will also become generally agreed upon.
Even though progress continues to be made, CRISPR could potentially not cut the correct genome section, or worse, cut out an essential section. To prevent this, we still need to dive deeper into the genome and its sequencing. Even though the research is hopeful and the outcomes are promising, it is likely to be a few more years before CRISPR-Cas9 is used regularly on humans.
Make sure to like and subscribe to our YouTube channel. We will be creating no new content each week. This week's subscriber shoutout goes to Miss Forney. This video was brought to you by Brooke Leach and Erica Schneider. Thanks for watching!